Hello everyone, I hope you guys are okay. So today we are going to have hematology in nursing 1.2. Under the topic of hemopoiesis, we shall check on the physiology and pathology of the same. So hematology is just but a subject or maybe a course unit that entails study of blood. Hem means blood, logy is the study. So when you talk about hemopoiesis, this is just the process where blood cells are made. The process by which the blood cells are made, called the hemopoiesis. When we're talking about gametes production, we mentioned gametogenesis, right? Spermatogenesis, oogenesis. So here we got hemopoiesis. So this is a process where the blood cells are made. The yolk sac and the latter, the liver and spleen, are important in the fetal life, but after birth, normal hemopoiesis is restricted to the bone marrow. So when the baby is still in the intrauterine life, the organs that helps in the production of blood are the liver and spleen. But after the fetal life, that is in the fetal life, but after that, now after the birth of the baby, what takes place in the hemopoiesis is the bone marrow. So the bloods are produced from the bone marrow and not the spleen or liver. Infants have hemopoietic marrow in all bones, but in adults, it's in the central skeleton and proximal ends of long bones only, normally told to hemopoietic tissue ratio of about 50:50. Expansion of hemopoiesis down the long bones may occur like when we have a condition like leukemias and the chronic hemolytic anemias. That one will now lead to the abnormal hemopoiesis tissue now working on, and then expansion of hemopoiesis, or the process of hemopoiesis taking place down even to the long bones. The liver and spleen can resume extramedullary hemopoiesis when there is marrow replacement. In case of chronic diseases like, let's say, leukemia, right, the myeloid leukemia, when we have bone marrow transplant in place, now remember, during that position, your body is no longer depending on the bone marrow for the hemopoiesis or the erythropoiesis. Blood production is now being shifted, and then the liver and spleen now assume that there is no option. Therefore, they come in as an alternative. As we proceed, I hope you are good. Now, for any inquiries, or maybe if you need to have a preview or maybe contact for any necessity, just Google Medical Students Arena. The details of contact information are there for the site. Is an online platform for learning. Now let's talk about stem cells and the progenitor cells. The common primitive stem cells in the marrow has the capacity to self-replicate, proliferate, and differentiate to increasingly specialized progenitor cells, which after many cells division within the marrow form mature cells, which we call the red cells, granulocytes, monocytes, platelets, and lymphocytes of the peripheral blood. So the earliest recognizable red cells precursor is a what we call the probromonoplast, and the earliest recognizable granulocyte or the monocyte precursor is the myeloblast. An early lineage division is between the progenitors for lymphoid and myeloid cell. Stem and progenitor cell cannot reorganize morphologically, so they resemble lymphocytes. The progenitor cells can be detected by special equipment in vitro assay. In vitro I mean outside the body, which they form colonies. Colony forming units for granulocytes and monocytes can be used to detect it, and then the stem and the progenitor cells also they can circulate in the peripheral blood and can also be denoted. So the stromal cells of the marrow, what we call the fibroblast, endothelial cells, macrophage, or the fat cells, they have adhesion molecules which react with the corresponding ligands on the stem cells and maintain their viability. Let's talk about the growth factors. Growth factors, just like any other growth hormone or anything that attribute to growth. So when you talk about the blood production hemopoiesis, this also is controlled by the growth factor. Just like how your body grows, it has to be controlled by the growth factor, which usually act in a synergy with each other. So growth factor and hemopoiesis will work in a synergy. When I say synergism, this is a thing that help another thing to, they help one another, right? Like your leg pushes, and let me talk about which one can I make an example for. Let's say your tongue pushes foods and your larynx swallows it, or maybe your pharynx swallows it. Yeah, so they are helping each other. That is synergistic. So both hemopoiesis and the growth factor enable each other to propagate the same function. So these are glycoproteins. These growth factors are glycoprotein produced by the stromal cells. For example, the T lymphocyte, also the liver also can produce some, on the for example, for erythropoietin, the kidney is the one which produces. Yeah, so I've said glycoproteins produced by either the stromal cells, the T lymphocyte, the liver, and the kidney, which now, the growth factor produced by kidney is what we call the erythropoietin. While some growth factors are mainly on the receptors on the surface of primitive cells, others act on later cells already committed to a particular lineage. They also affect the function of mature cells. So these growth factors inhibit apoptosis at most. So when I said apoptosis, to mean that they can inhibit death of the cell, which is supposed to be like is a programmed thing, but now they inhibit it of their target cells, that is. So growth factors in clinical use, the one which are used in our body, include the erythropoietin, which you've heard of it for greater time because the most which is used in production of red blood cells. So erythropoietin, another one, granulocyte colony stimulating factor, denoted by GCSF, or granulocyte macrophage colony stimulating factor, denoted by GMCSF, or the thrombopoietin, which is under trial currently. So those which I've mentioned are practically used in the clinic, in the lab right now for specific reason. For instance, somebody with anemia, we can attribute by injecting erythropoietin in the system and see how it can do its work. Yeah, similarly to those with a deficiency syndrome or chronic diseases and even clotting, coagulation problems or disorders of blood, we can inject them with thrombopoietin, which will initiate the production of thrombocytes, the platelets. So away from all that, let's take on signal transduction. Just like how receptor behaves, the binding of the growth factor with its surface receptor on the hemopoietic cells activates a complex series of biochemical reactions by which a message is transmitted into the nucleus. So the pathways involve sequential phosphorylation of substance by protein kinases, and then this signal activate transcription factors, which in turn activate or inhibit gene transcription. The signals may activate pathways which cause the cell to enter cell cycle, that is replication cycle, differentiate, maintaining viability by not dying or inhibiting apoptosis, or becoming functionally active, and therefore enhancement of cell killing by neutrophils. So what I'm trying to say that the effect of the growth factor to initiate hemopoiesis, something like the growth factors inashikana kwa receptor kwa hizo hemopoietic cells, and then they start on activating the complex biochemical cascade which now react, and this one will involve sequential phosphorylation. And I think according to your knowledge of endocrinal system and the nervous system, how communication between the ligands and the ion gate takes place, we have phosphorylation, phosphorylation cascade where phosphate is added, and therefore by this there is signal for transmission of various factors. So it will initiate that growth, division, and then if it is red blood cells, those cells will start by the progenitor cell dividing as we attribute over to getting what we are looking for, that is the blood cells. Assessment of hemopoiesis. How do we assess that the blood is progressively produced? Hemopoiesis can be assessed clinically by performing a full blood count on peripheral blood. So you'll do the PBF and then you literally do a full blood count. Nowadays, full blood count is very, very vital for many necessitated conditions. So it is an indication of variety of clinical conditions. Then we can also use bone marrow aspiration. Also, it allows assessment of the latter stages of maturation of hemopoietic cells. So when we do aspiration of bone marrow, we can check on the component. I told you normal bone marrow should have a pressure of 50 to 50 to 500 millimeters of water. You need to know how it looks like, the color, the appearance, the smell, and so on and so forth, the density, right? So that is that. We have a trephine biopsy, provides also assessment tool. Trephine biopsy provides a core of bone and bone marrow to show architecture, so that one also can be used. So whatever you're able to see there, no, they are very hardly visible from your hand, but as much as you can hear me talking about them, we have action on stroma. That is now how the growth factor takes place. We have the interleukin 1 together with the tumor necrotic factor. Tumor necrotic factor will link or combine or infuse with interleukin 1 to cause or stimulate production of the GM-CSF, which I'd already told you. Yeah, I'd given you the name and the description of those attributes. The one which I told you, we have granulocyte and monocytes. So all that one, and after that we have action on pluripotent cells of the stem cell factor. When it acts on early multipotent cells, it produces the intermediate metabolites like interleukin 3. These are just the growth factor. So when they play their role well, we end up having blood being produced. So the same diagram presents here. You can see how the differentiated cells compartment, how they look like. Yeah, from the pluripotent cell we have the stem cell factors. We now give us the precursor, the GEM or the GEM, which will produce the granulocyte, monocytes, and then we'll have interleukin 3. So all of them we have B cells produced from the line for progenitor, thymus, and T cell as well. So we'll have the erythropoietin from that is responsible for production of red blood cells, thrombopoietin, which is now for thrombocytes, the granulocytes, CSF, the granulocyte macrophage, the granulocyte monocyte, the leukin, the interleukins, all that one, the leukocyte 5, interleukin and interleukin. So all that one are very, very key. So away from that, we have similar diagram presented below, and you can analyze this one, have pause, check on what happens, and you can also do your own discussion on the basis of what you see. That is now peripheral blood smear, which you can use to have the full blood count. And then we can also roll in and check on the hemopoiesis processes. So from there, we are supposed to start on the normal blood cells, the red cells, and the platelets. Now just to mix them together so that at least we can figure out what we'll do next. Peripheral blood cells are very key. The peripheral blood cells, they are normal peripheral, contain many mature cells which do not undergo further division. They are the blood cells which are found on our limbs, on our body part, which are outside. So we shall talk about the red blood cells, we shall talk about the platelets, we shall talk about all these ones, but now in the next hematology class. For now, let's end it from where we had hemopoiesis and physiology and pathology of the same.